All right. My first paper on this was in 1994. It came about in an interesting way. At that time, I was interested in the use of molecular markers for estimating relatedness. And I was thinking, okay, how do I know, how do I show that markers are superior to pedigrees for estimating relatedness? And I go, okay, for me to do that, I would need to come up with, to apply that to some procedure in which you actually use that relatedness for making genetic evaluations. And then I know, oh, okay, my animal breeding colleagues were talking about BLUP, and I know you use pedigree relatedness. I think we can plug in just marker relatedness. And if the marker relatedness gives a better prediction, then I said, then that would be proof that markers are better. And so that's how I got started. It was in a bit of a roundabout way. But it does point to two features of a plant that make BLUP useful in a breeding program, and these are straight from the text. One is it helps us handle unbalanced data. Although breeders tend to conduct experiments, individual experiments that are balanced, meaning that you have a set of lines or hybrids or test crosses or clones, and you evaluate them in the same locations in a given year. Chances are, yes, you might have missing data here or there, but for the most part, that these are balanced data. However, a natural feature in breeding is that the superior candidates are kept for further evaluation and new candidates are evaluated. And when you look at the totality of the phenotypic data available in a breeding program, the data are very highly unbalanced. A second feature that makes BLUP useful is the use of information from relatives. Typically in plant breeding, we evaluate a candidate based on the performance, the phenotypic performance of that candidate by itself. However, in the animal breeding literature, because you cannot replicate a cow or a pig, they're used to extracting information from relatives to come up with more precise predictions of genotypic value. And again, the classic example of information from relatives is in dairy cattle for evaluating the milk yield of a dairy bull. A bull does not produce milk, but it has genes that affect the milk yield performance of its female progeny. And so therefore, the milk yield of a dairy bull is evaluated on the basis of the performance of its relatives. So these are two the main features that make BLUP, best linear unbiased prediction, particularly useful in a breeding program. And when we talk about BLUP, we also talk about BLUE, best linear unbiased prediction versus best linear unbiased estimation. And in estimation, we were saying that these are for fixed effects. So we create a linear model and some effects would be fixed and some would be random, whereas prediction, this would be for random effects. And so in random effects, as we have discussed earlier in the class, there is a covariance structure among these random effects, meaning that, for example, if we're looking at breeding values, the breeding values of the different individuals are correlated if they are related with each other. General combining ability, specific combining ability, those are correlated with each other among relatives. So if we have random effects, there's a covariance structure. With a fixed effects, these are constants. And so there is no constants with no covariance structure. And typically we think of these as nuisance factors that we need to account for. They could be levels of fertilizer, they could be transgenes and so forth. Yes, Flutur, you had raised your hand, you have a question?
Yes, about the unbalanced.
Yes.
Does that mean that I could, for example, if I have, I don't know, a set of 200 lines and I test them in 1 year, but in 2 locations, and then the next year I test them again, but only in 1 location, and then I would still be able to calculate them or to get to use all 3 environments. Because usually I would probably then calculate them by themselves, like divide it into the locations maybe.
Yes, although if this is the case, if you have 200 lines in year 2 in 1 location, in a way, this is still balanced if you count, if you're looking at years and locations. Now, if I consider these as 2 environments, okay, and this is 1 environment, actually I would consider these as balanced data. What we're talking about is if in, say, year 1 you had 200 lines in 2 locations, and in year 2, let's say you kept 50, okay, and you evaluated those 50 lines, but then in addition you evaluated another new 75, okay, then this would be unbalanced, because now I have 275 lines, okay, total, but 75 only in year 2, location 1, and then, and then the rest are, 50 would be in all 3, and 200 in just the first year. So yes, this would be the unbalancedness that we are talking about.
But then, so if I have different, maybe if I test then 200 lines in year 1 and 2 location, but in the second year I test only 150, then that is what you would consider unbalanced.
Yes.
Okay.
Yeah, that would be correct. Yeah. Okay.
Thank you.
Other questions before we proceed? It's a very good question. What exactly we mean by unbalanced? Okay, and then finally, this linear model underlying BLUP and BLUP is called a mixed model, simply because we have a mixture of effects that are considered as fixed, and we have effects that are considered as random, and therefore this is a mixed model. And we'll see then that in the mixed model equations, for the fixed effects, there is no covariance structure that is specified. Whereas for the random effects, there is a necessity of we need to add a covariance structure for these random effects. Otherwise, and that really is the only difference, is that you're adding a covariance structure. In fact, the BLUP and BLUP methodology, if you're familiar with multiple regression and matrix notation for multiple regression, then the mixed model equations has very strong similarities to what you have for traditional multiple regression. Okay, any questions, comments?
Question from my side.
Yes.
In the textbook to BLUP, you write, in plant breeding, an individual environment is considered random, but the mean of a set of environments used in performance tests is considered as fixed. Can you explain? Because since one environment, how can it be random? Where's the covariance structure?
Oh, for the random environments. So that covariance structure then for those random environments, that has to deal more with the inference space. And by the way, this covariance structure, it is possible that it could be simply an identity matrix, meaning that it could be a 0 covariance, but first of all, it can be a 0 covariance with other environments, just to make that clear. And it can be an identity matrix. And oftentimes, and that's what we see for the random errors, for example, that is, it is an identity matrix. And so the random environments assumption has to do more with the inference space. If we have random environments, we're saying that the inferences apply to the population to the target population of environments, rather than only to those environments that were sampled. However, the point being that when we take random environment 1 and random environment 1 and environment 2 and environment 3, okay, there would then could be a mean associated with these first set, okay? And then when we take another set of random environments, environment say 4 and environment 5, and we consider that as another set, there could be a mean associated with that other set. Meaning that if environment 1, 2, and 3 happen to be, say, in a wetter year, and environment 4 and 5 happen to be in a drier year, then the mean of set 1 and set 2 would be different from each other. And we need to account for that. However, we're still saying that even though it just so happens that environment 1, 2, and 3 are in a wetter, we still consider all 5 environments as representative of the target population of environments. So it has to do with more with the inferences that we make as to what environments are we applying these inferences. Yes, Francisco.
I have a question. How are you able to compare between BLU's means or the value that you get for each line, like a Tukey test or Duncan, or you just get a value and then you said it's bigger than the other one? Or there's a way that you can just check for significance between?
For BLUE, yes. For BLUE, yes. But you don't do a Tukey, or well, you could, say do a t-test or a z-test, because the best linear unbiased estimates, they would be, if sorry, jumping to notation here, there would be a variance associated with the sampling variance associated, and so you could use this in statistical tests. So yes, we do statistical tests with BLUs, but not with BLUP. So basically, what you get in BLUE is identical to what you would get with a generalized least squares procedure. And so, to the same extent that you can do statistical tests with fixed effect estimates from generalized least squares, then you could do this in that context as well.
Thank you.
You're welcome. All right. Was there one more question? Let's see. What if all factors in the models are random? What will be the fixed factor in the mixed model? Yes, it will just be the overall mean. If all the factors are random, there still is the overall mean, and that would be the only fixed effect. You're welcome. Okay, so what I'd like to do now is go and construct an example of how the mixed model equations might look like in a small data set such as this one. Okay, so you have this handout, and so I'll be writing some notes, but I'm going to encourage you to come alongside me and try to see if you could come up with what we come up together. And so let's take a simple example. And the nice thing again with matrix notation is that what works out with a small example would also work out conceptually with a larger, and computationally with a larger example. So suppose a soybean breeder develops inbred 3 from the cross between inbred 1 and 2. So you have parent offspring relatedness. We're assuming inbreds 1 and 2 are unrelated. And then the coefficients of coancestry, we're just looking at pedigree records. Between 1 and 2 are 0 because they're unrelated. Between parent 1 and the offspring is 0.5. And between parent 2 and the offspring is also 0.5. We're assuming that the inbreds 1 and 3 are evaluated in a set of yield trials, 4 locations in year 1, and we're calling that a set number 1. And then look, inbreds 2 and 3 are evaluated in a different set of yield trials, say set 2 at 4 locations in year 2. Okay, so you have 4 locations in each case. They're different years. We're just using same number of locations to make things simple. They don't have to be the same number of locations. And the results of the yield trials were as follows. You have trial 1, 2, 3, 4, the inbred line, and then the yield of that soybean line. And so in scalar notation, scalar being just the usual numbers, individual numbers that we are familiar with, a reasonable linear model would be something like this, where Yij is the yield in set I of inbred J, okay. Mu is the grand mean. Ti is the effect of set I, meaning that you had 2 sets of yield trials. They could have different means associated with those 2 sets. So we have a set, the effect of set 1, set of trials, and set 2. And then the Uj, that would be equal to the effect of the ith inbred. And then Eij is simply the residual term. In this case, because we're looking at the means, because the individual observations are the means across environments or across locations, there is no explicit genotype by environment interaction term. However, this residual term includes a portion of your within error variance, which is confounded with your genotype by environment interaction variance. So there is G by E interaction. It's just confounded here in the residual term because of the way we expressed the data. All right.
Question.
Yes, go ahead.
Do you check? You see, I have the problem with the residual term. Sometimes, do you need it to reduce to white noise? Do you check for that? Do you detrend the G by E interaction out? Or is a general term where you gather everything, residual, white noise, in the purest sense, plus all the other interactions? So then why don't you have epistasis and things like that? That's the
why we don't have epistasis.
Residual.
No, so this is generic. It's so in this case it is whatever is not accounted for by the other terms.
That's okay, but how do you know? Because we partition the variance into different things, but how do we know that we don't have a epistasis in there?
We don't know. We don't know that there is none.
A priori.
Yeah, we don't know that there is none. Yeah. I mean, if we wanted to include epistasis in the model, we can. We can do that. But we're assuming for this purpose here that it is not present, and so residual is a catch-all that simply says the
leftover.
It's a leftover. In this case, though, we need to specify a covariant structure for the leftovers. So in this case, we actually need to make genetic assumptions that we're not modeling epistasis. Okay. All right. So if we now take that familiar linear model, I'm going to fold my sheet here so that I have more real estate in my desk. So that familiar linear model, let's see, there you go, can be expressed in matrix notation as follows. You have Y, it's a vector, is equal to an incidence matrix called X multiplied by B, my beta. Okay, and then plus Z, an incidence matrix, times U plus E, and I'll write down how these all look like. So my Y is just my vector of observations. So I see here that I have 4 observations, 20, 25, 40, and 30. And so therefore my Y is simply that. I have 20, 25, 40, and 30, okay? This X matrix here, well, let me write down the beta first. I'm going to put 2 factors under beta, and I'll say that's B1 and B2, where I will define B1 as equal to the mean plus the effect of the first set of yield trials, and B2 as the mean plus the effect of the second set of yield trials, okay? Meaning that, yes, I have 2 elements in my beta vector. The X then would relate each observation in Y to what I have here in beta. So beta has B1 and B2. We need to devise a way of relating each observation to the specific effects here. In this case, the incidence matrix would have 2 columns. It would have 2 columns because there are 2 elements in beta, and it would have 4 rows because there are 4 observations that we need to tie to this B1 and B2. And so this 20 over here was for an inbred that was evaluated in set 1. So therefore I would put 1 and then 0. The 25 was also evaluated in set 1. So that would be 1 and 0. Okay. And then the 40 was in set 2. So I'd put 0 and 1, and then 0 and 1. So the way, just to think of this, is we're saying that the first column here refers to set 1, and the second column here refers to set number 2. All right. Questions, comments? Is that clear so far? All right, let's then construct U, and these U are for the inbred lines. There are 3 inbred lines, so this one would then have 3 elements, which we'll just say U1 for inbred 1, U2 for inbred 2, and U3 for inbred 3. The Z, the incidence matrix, would again have 3 rows. The number of rows is the same as the number of observations, and it would have 3 columns because there now are 3 factors that we have here in mu. And so the elements of that, the trial 1, I mean the first observation of 20 was for inbred 1, so therefore that is 0, means 1, 0, 0. The second element of observation of 25 was for inbred 3. So we write here 0, 0, 1. And then 40 was for inbred 2. So therefore we put a 1 in the second column and zeros in the rest. And finally, the third one of 30 was for inbred 3. So therefore we put 1 in the third column, 0 and 0. So again, the way to look at this is the first column is for inbred 1, the second column inbred 2, and the third column is inbred 3. All right. That clear so far? Any questions? Pretty simple so far. And then we just to complete it, we have our residual terms, and we will just denote that as E11, E13, first subscript is for the set, the second subscript is for the inbred line, and then this is E22 and E23. All right. So again, all we have done here is express this familiar scalar notation that we have in matrix form, and then we just started to build these matrices or write down what they would look like. Okay. Questions, comments? Is that clear so far? Okay.
Correct.
And yes, go ahead, Francisco.
Just a short question. If you want to get the blues instead of the BLUPs, you have to put down the B1, B2, then you have to include U1, U2, and U3, and it's the same, like the same idea, mu plus U1. If you want to get the blues?
For
the genotype, yes.
Yeah, if I want the blues here, I think I would just not put, I would not add the mu's. I think I would just put mu1, mu2, and mu3.
Okay.
Because the restriction, basically the restriction that we're having is that B1 and B2 sum to 0. It's basically the restriction. B1 and B2, if we add those two, they sum to 0. And because we already accounted for that sum to 0, if you want the blue instead of BLUP of your muse, you don't need to account for that a second time.
Okay.
Okay. So what we have here then is that we have the beta is then a vector of fixed effects. U is a vector of random effects. Then X and Z are just incidence matrices of ones and zeros relating X to B and Y to U. This is jumping ahead, but sometimes if you're looking at marker data and you need to do some sort of imputation and you're not quite sure whether it's this marker or the other marker allele, your incidence matrix might not be a 1 or 0. It might be 0.8 and 0.2. If you think there's a 0.8 chance that it is marker allele 1 and 0.2 chance that is marker allele 2. So typically the incidence matrices are 1s and 0. However, if you need to do some sort of imputation, then you could use proportions or fractions in your incidence matrix. All right. With regards to distributions, we're assuming that the UJs are distributed with a mean of 0. They have a mean of 0 and a variance of G. And then the elements of the G matrix then depend on the type of genetic effects. In this case, we have inbred lines, so it would depend on the effects, genetic effects expressed among inbred lines. If we are looking at clones, let's say that you're a breeder of apple or a sweet potato and you have heterozygous individuals, then that would need the G matrix would reflect that. If you're working with single crosses, actually you'll have more than one genetic component that you would have to model. But in this example, the relatives in question are an inbred and its two parental inbreds. We're assuming dominance variance is 0, but not only that, because they're inbred lines, dominance variance is not expressed among inbred lines. And so we're saying that the covariance of between inbred J and J prime is equal to twice. This is the coefficient of coancestry. And then VA is the additive variance. And we are expressing then G as equal to A times VA. That means to say that the elements of A are equal to twice the coefficient of coancestry among the individuals. So therefore, the G matrix in this case is equal to, again, we have now 3 inbred lines, so we need a 3x3 matrix. The coefficient of coancestry of an inbred line with itself is equal to 1. And we need to multiply that by 2. And so therefore we have 2 in the diagonals. And then so let me before I write that down, what we're basically saying here is, again, the first row, the first column is for inbred 1, the second is for inbred 2, and the third is for inbred 3. And we're doing the same here. Again, the first row is for inbred 1, the second row is for inbred 2, and the third row is for inbred 3. And so now what we're saying is that this element here would be the covariance or the coefficient of coancestry, or twice of that, between inbred 1 in row 1 and inbred 2 in column 2. And we said that the two parents are unrelated with each other. Inbred 1 and 2 had a 0 coefficient of coancestry, so 0 times 2 equals 0. And then for inbred 1 and inbred 3, it is a parent-offspring relationship. Their coefficient of coancestry was a half. We need to multiply that by 2 in this case, so that becomes equal to 1. And then between inbred 2 and 3, again, same parent-offspring relationship, coefficient of coancestry of a half multiplied by 2 equals 1. All right. It is a symmetric matrix, and so this would be also 0. Okay, we interchange the rows and columns. It's the mirror image, so to speak. And so that's what the A matrix would look like. And to get the G matrix, you need to multiply each element by your estimate of VA. All right. Questions, comments? Okay. The expected value, the expectation of your Y vector is equal to the incidence matrix times XB. Some distribution wise, we're assuming that the residuals are distributed with a mean of 0. Again, with random effects, that's a typical assumption. And a variance that, just like the variance of genetic effects is G, is A times VA, the variance of your errors is equal to R times VE. And if we assume that there's a common homogeneous error variance, then R is an identity matrix. And we can assume that in this case, if it's homogeneous plus each observation was obtained from 4 locations, because set 1 had 4 locations and set 2 had 4 locations. Otherwise, if set 1 had a different number of locations from set 2, then it's no longer an identity matrix, but we need to account for the difference in the number of observations in each that went into each element in Y. And so it becomes equal to 1 divided by the number of observations. But we don't need to worry about that in this case. The textbook does give an example of how that might look like if you have an equal numbers of observations. Questions, comments? That clear so far? Okay. Now we will then put together what are called as the mixed model equations that we would then need to first put together and then later solve. So we actually won't be doing any solving because we have a larger number of equations than we can conveniently solve by hand. However, I think as a pedagogical or teaching exercise, it's still helpful to go through how we set up these mixed model equations. Some of you might have been in seminars where you have seen mixed model equations and you freak out and get confused. There's no reason to be confused. It's not that hard, and we're going to show that here in a few minutes. So the mixed model equations are as follows. You have your solution vector, which has 2 components. It would have the estimates of my fixed effects, and it would have my estimates, my predictions of the random effects. Okay, that would be my solution vector. And this then is equal to, I'll just write this down and explain it later on, x prime r minus 1 x, okay, and then x prime r inverse z. I'll have my Z prime R inverse X. And then I'll have my Z prime R inverse Z. And then I need to add inverse of my A matrix times the ratio between residual and additive variance. Okay. That's what I would call the coefficient matrix. So this is the solution vector. This here is the coefficient matrix. Okay. You need to take an inverse of that, kind of like division. And then you multiply that with what is called then as the right-hand side, right? For lack of a better term, vector. And this is X prime R minus 1 Y and Z prime R minus 1 Y. Okay. And what we'll do today is actually build all this up and go through, okay, exactly what does this sub block here look like? What does this look like? What do each of these look like? That's where we're headed in today's class. Now, if R, yes, go ahead.
In what does it differ, this formulation, from Henderson's model, from the 75 model?
It's the same.
Okay, so it's,
yeah, it's the same.
So far you didn't contribute that much.
Yeah, it's the same. Yeah,
I mean,
yeah. Okay. So if R is equal to an identity matrix, then it simplifies here because the inverse of an identity matrix is an identity matrix, and it's like multiplying it by 1 or dividing it by 1. It doesn't change a thing. So then we have B and then U. Okay. So we just drop out the R inverse. So we have X prime X, X prime Z, Z prime X, Z prime Z plus A minus 1, VR over VA. We take the inverse of that, we multiply it by X prime Y and Z prime Y, okay? All right, so just a little segue. If you're familiar with linear regression, multiple linear regression, in multiple linear regression in matrix notation, your regression coefficients B are equal to X prime X. You take the inverse of that and you multiply it by X prime Y. And so you see just strong foundations here with multiple regression because I have my beta here, okay? And then I have my X prime X here. And then I take the inverse of that and I have my X prime Y. So we see here a strong similarity between what we've just set up with multiple regression. It's just that instead of everything being fixed, we have U, which are random effects, and therefore we need to add this portion here that captures the covariance among these random effects. You'd see then that if these were fixed rather than random, then this entire term drops out, and then it's multiple regression again. U equals Z prime Z, inverse of that, Z prime Y. And so it's just nice to think of how those two relate with each other. Okay, so the submatrices, we'll begin to build them. Let me see if I can still find my previous sheet of paper here. Where is it? Here it is. Okay. So we built our X matrix here, and now we can begin to build these submatrices. See here if I can, there you go. Let's start with X prime X. X prime is equal to the inverse X prime X. X prime is equal to, not the inverse, the transpose of X. We just exchange the rows and columns. So if X was 1, 0, 1, 0, 0, 1, 0, 1, we just flip it to its side, if you would. And so our X prime would be equal to 1, 1, 0, 0, all right? And then 0, 0, 1, 1, okay? That's our X prime. And then our X is what we had written earlier. It's this one, 1, 0, 1, 0, 0, 1, 0, 1. All right. Are we together? Again, this is just X. This is X prime. You'll see that if this were the X matrix, we flipped it in this way, so to speak. Okay, we flipped it like that. And then we get our X prime. And then we need to multiply these two. Just of note, you can multiply two matrices that we have here, a 2x4. Our X prime is a 2x4 matrix, and our X matrix is a 4x2. And these two need to be equal to each other for them to be multiplied. And in the end, what it tells us, we would get then a 2x2 based on these dimensions over here. These would give a 2x2 matrix. And so how we do that, we in matrix multiplication for this first element, for row 1, column 1, you take the first column of the first matrix and you multiply each element with the elements down the first, I'm sorry, let me say it again. You take the first row of that first matrix and you multiply and sum with the elements in that first column of your second matrix. In other words, it'll be 1 times 1 plus 1 times 1 plus 0 times 0 plus 0 times 0. Let me say that again. I'm taking this element 1, then I multiply it by 1, plus 1 multiplied by 1 going down, plus 0 multiplied by 0, and 0 multiplied by 0, and that gives me 2. Then for row 1, column 2, I take row 1 in the first matrix, column 2 in the second matrix. I repeat the same. 1 times 0 is 0. 1 times 0 is 0. 0 times 1 is 0. 0 times 1 is 0. So that gives me 0. I can do that same operation for here. I would get 0. And finally, for the last one, it'll be then row 2, column 2, that would be 0 times 0 is 0, 0 times 0 is 0, 1 times 1 is 1, plus 1 times 1 equals 2. This then is my X prime X matrix. Questions? Yes, go. Yeah.
This is a symmetric matrix, and yet you do the second 0 calculations as much as you did before. So why don't you take advantage of the symmetry condition and you don't even calculate? But that's a side comment. The main question is when this matrix is non-symmetric. Under which conditions the matrix is non-symmetric?
Yeah, and so yes, to your point, yes, it would be symmetrical, and if it doesn't really change that much, it's easy to calculate. Whether it'll be non-symmetrical, I don't think it would be. It should be symmetrical.
Yeah, the question is not even when you have the marker allele broken down into 0.8 and 0.2. always the matrix will be symmetrical even for the not 1 but 0.8
so
and
yeah
0.2?
so that it might be it would be asymmetrical, I think, in that. Well, what would happen in that case is that you would have a non-zero element here.
Okay.
I think
Well,
it would
and
still
then,
be
and then the other
I think
zero
it would
will,
still be symmetrical, yeah, and a non-zero element here, of course. Yeah.
okay.
Okay. All right. And so what I'll do now is we calculated this first part. I'm sorry, x prime x. We'll now do x prime z. Let's see, I need to keep track of all the papers I have here. Let's see, I need to keep track of that one. I usually do this in a blackboard. Okay, so why don't I let you do this as a bit of seat work for yourself? X prime z, okay, and then z prime x, and then z prime z. Why don't you spend a few minutes trying to figure those out, and then we'll do that together. Okay, has anybody gotten x prime z? Raise your hand if you have.
Is 2, the product, the matrix is 2 product 3. I don't, how do you
It
say
is
that?
2 by 3. Yes, it is a 2
2
by
by
3.
3, yes. It's 1 0 1 0 1 1.
Correct. So yes, let's write this down. So your x prime, which is the same as before, in the previous one we worked with, was 0 1 1 0 0 0 0 1 1, and then the z matrix was yeah 1. I'm sorry. It's S3 1 0 0 0 0 1 0 1 0 and 0 0 1. Okay. And if you do the matrix multiplication for those two, you would get 1 0 1 and 0 1, 1. All right. All right. Your Z prime X is actually, because it's symmetrical, your Z prime X will just be the transpose of what we have just obtained. So your Z prime X would be 1, 0, 0, 1, and 1, 1. I mean, you could write it out, do the same exact, and I'll show that in the notes. But again, it's just a transpose of this one. All right. Anybody got in Z prime Z? Nobody yet? Okay, let me wait a little bit. No. So the way
3 by 3 matrix.
Is it a diagonal? What are what's the diagonal? What are the diagonal elements that you got? Did you get that?
1,
yet?
1, 2.
That is correct. It's 1, 1, 2. And so just to write that out, Z prime is equal to 1, 0, 0, 0, 0, 0, 1, 0, 0, 1, 0, 1. And then Z would be 1, 0, 0, 0, 0, 1, 0, 1, 0, 0, 0, 1. And if we multiply those two, we would get a 3 by 3 matrix, which should be 1, 0, 0, 0, 1, 0, 0, 0, 2. All right. So let me explain this here a little bit. If we go back to our X prime X, basically what you have in the diagonal elements is this one here is the number of observations in set 1. And here it's also the number of observations in set 2. All right. Each set had 2 inbreds evaluated in it. And likewise here for Z prime Z, this is the number of observations, for inbred 1. And then this is the number of observations for inbred 2. And finally, this is the number of observations for inbred 3. We notice that in the data set, inbred 3 was evaluated in both sets 1 and set 2. Therefore, it had 3 observations. All right. Questions? That clear so far? Okay. Now, both VE and VA are unknown and they need to be estimated. I think I used VR, I'm sorry. Both VR and VE are unknown and they need to be estimated. But now, just for now, let's assume that we actually know the ratio of them. VR, VA is equal to 0.25. The A matrix, we said the A matrix is equal to twice the coefficient of coancestry. So 2, 0, 1, 0, 2, 1, and then 1, 1, 2. We obtained that earlier. If you obtain the inverse of this one, A inverse, I'm just going to write it down. I used a computer program to do this. That's 3/4. That's a 1/4 minus 1/2. 1/4. 3/4 minus 1/2. Okay. All right. And again, we put this because yeah, we need that result later on to put together the mixed model equations. So moving on, we go to the right-hand side of the mixed model equations. The right-hand side is x prime y. And so x prime transpose is 1, 1, 0, 0. And then we have 0, 0, 1, 1. Okay. And we multiply that by the y vector.
Sorry, correct. The
Yeah.
inverse of A, we
Yeah.
we can calculate, or you calculate with a program.
I used a program for this one. You could, with some difficulty, a 3x3 by hand, but once you get, again, in the class, in the book, we stuck with a 2x2. Beyond a 2x2,
Yes.
we just say, okay, we're going to use software to do that. Yes.
Okay. Thank you.
All right. And so the elements here are 20, 25, 40, and 30. All right. And if I do that, if I multiply these two matrices, what I get are 40 and 75. Okay. You would notice that the 40 and 75 are simply the totals for each set. Meaning that if we take out our sheet here of data, if you add the first two, I mean, the 40, I'm sorry, it's 45. You write that down. It's 45 and 70. 45. Okay. Sorry about that. It's 45 and 70, meaning that if you take the total of the first set, you get 45. If you take the total of the second set, that is 70, and that's what we get here, okay? And likewise, we get z prime y, and z prime y is equal to, I'll just write down z prime, 0, 0, 1, 0, 0, 1, 0, 1, okay? We multiply z prime by the same observation vector of 20, 25, 40, and 30. In the end, we would get 20, 40, and 55. And you notice that these are the totals for each inbred. Okay, meaning we look at our data set. Inbred 1 was evaluated once. It had a yield of 20, so therefore we have 20. Inbred 2 was evaluated once, had an observation of 40. We therefore have 40. Inbred 3 was evaluated twice. We add those two observations, 25 and 30. We get 55, and so hence that's what we get there. So point being that even though this might look, they could look complicated, in the end, the numbers actually have that rhyme and reason, or that sense to them, okay? So therefore, by now, we have built all of the needed submatrices to put together the mixed model equations. And here's what they would look like then. B mu should put much space here, sorry. This is 2 and 0 and 0 and 2. Okay, this was, just use this to say that's the x prime x. Okay. And then we had calculated x prime z as 1, 0, 1, and then 0, 1, 1. Okay, that was the second block, so to speak. And then we have the transpose of that, 1, 0, 1, and 0, 1, 1. Okay. And finally, we need to take what we had in our z prime z. Sorry. See my paper now. Okay, here we go. We take what we had. First, we need to multiply the inverse of A by the ratio of VR by VA, which we assumed is equal to 0.25. So basically we get a quarter of each of these values, and then we add that to z prime z over here. And so when we do that, and you can verify this with the notes that I will send later on, what we're left is in fraction form. I'll just put 1 and 3 over 16. 1 over 16, negative 1/8. Write that down, 1 over 16, negative 1/8. Okay. And then again, 1 and 3 over 16, negative 1/8, negative 1/8, and finally 2 and 1/4. We'll need to take the inverse of that one, and then we multiply it with the right-hand side, which we had obtained here as 45 and 70, and then 20, 40, and 55. Okay. And again, this is, again, you use computer software to do that. Typically, it's a large set of equations. But in the end, if you do solve those sets of equations, what you would come up with is that our beta, our estimate of beta and mu, our solution vector is equal to the following. It's 26.5 and 33, and then -6, and then 6 and -2. Okay. In other words, what we're saying is that this 26.5, we said that was equal to the mean plus the effect of the first set. The 33, that's equal to the mean plus the effect of the second set. This is the effect of inbred 1. I mean the BLUP of inbred 1. This would be the BLUP of inbred 2, and this would be the BLUP of inbred 3. All right. Questions, comments? Questions or comments?
I have a question.
Yeah, go ahead.
Yeah, we use, -1 and the -4 inverse. What is the difference between the two?
Okay, so the -1 is what's, so then just the negative, the minus sign, that's called a generalized inverse. The -1, that's an inverse. So sometimes you might come across a set of equations in which there is no unique inverse. There is no unique solution. However, there exists an infinite number, theoretically infinite number of what's called as a generalized inverse. What happens is that sometimes, typically, in the fixed effects, that they would not be a unique solution to the fixed effects. And that happens because each column in a matrix is not independent from each other. Let me illustrate, for example, how that can happen. Let's say that we want to estimate the grand mean and the effect of set 1 and the effect of set 2 and the effect of set 3, okay? We want to separately estimate the grand mean. The incidence matrix might look something like this. We could have we fit the mean, okay, for each observation. Let's say we have only one observation for each, okay? This is the mean, and then T1 would be T1. Sorry, just take that out. 0, 0. And then the second observation would be for T2, so 0, 1, 0. And the third observation would be for T3, 0, 0, 1. You would notice that the sum of these three columns is equal to 1, and that sum of these three columns is all equal to what you have in the first column. In that case, you have dependency here. You have dependency in that the columns or functions of these columns are not independent from each other. And so in this case, if you have something like this, actually you will not have a unique inverse. And so you'd have to resort to what is called as a generalized inverse to come up with a solution. The point being that even though the solutions of U and T1 and T2 and T3 are not unique, however, U plus T1, U plus T2, U plus T3, each of these remains unique. Each of this is estimable. And this is what is called an estimable function. Meaning that even though we cannot we cannot uniquely estimate each of these four, that each of these functions is actually estimable. And so we use then a generalized inverse to come up with a solution. It's not the only possible solution, but to come up with a solution and to be able to still have estimable functions of these fixed effects. But yeah, that's a long answer to a generalized inverse versus an inverse, but that's basically, again, when we have just that, then we have a generalized inverse, and when we have -1, then that is a unique inverse. Okay, I don't know the text numbers, the page numbers, but we talked a little bit about that in the textbook. Yeah. Other questions?
Yeah, I have one more question.
Go ahead.
In the solution, we have, the values for the first two for B and the last three for U. Can we say, the first two as blue and
yes,
the other?
Correct. This is the blue. And these three are the BLUPs. Yes. Yeah, these are the blues. Yep, correct.
Okay, thank you.
You're welcome. Other questions? All right, I have a question for you. We said that the expected breeding value, or the mean of breeding values is equal to 0. Or the expected value of the mu's is equal to 0. How come that's not the case here? You take -6 and 6 and 2, and that's not equal to 0. Any thoughts?
Probably by multiplying with the frequencies.
No, yeah. That's somewhat correct in the sense that Yes, actually, when we say the mean of breeding values is 0, that refers to the base population. And so if you take the two unrelated lines, parent 1 and parent 2, the mean of their breeding values here is actually 0. But once you add more individuals, more lines for which there was some, say, selection going on or drift or whatever other, it's no longer in the base population, then the mean is no longer equal to 0. Yeah. All right, we're past time here, so I'm going to formally dismiss the class. We'll pick up this discussion next time, but I'll remain in the class for a few more minutes in case you have questions that you'd like to ask about this exercise we just did. Francisco, yes. Yeah.
I have a question related with the, well, thank you for saying that the mixed model is difficult. I have been in other classes and it's not easy to understand at the fourth time that I'm taking. So can you go to the page where you have problem 2, exercise 3, where you define that maybe it's not the scope of this part, but it's the error variance.
Okay.
When normally in some papers or when you are trying to understand, they define that the error variance of this mixed model, and they can also like adjust the error variance, like for the correlation between, I am thinking about G by E, or maybe there's any factors in the environment when you are testing and you are adjusting that error variance. So could you just give an example of
Sure.
how that works?
Sure. So let's say that so typically we said that the covariance structure of your errors or your residuals is equal to R times VR. In this case, in our example, because we had equal numbers of locations in each set, we simply said that our R then is equal to an identity matrix, okay? And they're all zeros in the diagonal. Now, let's say that we have set 1 has 4 locations and set 2 has 8 locations. Then the R matrix, we need to account for that by now saying that we no longer have an identity matrix, but then we would have a fourth, an eighth, and an eighth. And when we do that, the VR is on a per location basis, and that would be slightly different from the VR actually we'll have here, because this would be the VR associated with the mean. Okay, it's a VR associated with the mean. This is a VR associated with each observation. And so typically in this case, if we're accounting for different numbers of observations, the covariance structure, the off diagonals would be 0. There would be no covariance. Now, however, let's say that you're explicitly modeling genotype by environment interaction, and you would say, oh, in location 1 and in set 1 and in set 2, there's actually some correlation between them. They have, same soil type or whatever it is. We apply the same cultural management practices. Then you could have a non-zero. You could have non-zero diagonal elements. I mean, non-zero off diagonal elements to express the correlation among those factors. So in the same way that for the genetic effects, we can express a covariance or correlation, in the same way for non-genetic effects, if we have information on the non-zero covariance, then we can include that in the model.
So it's the same idea as the
It's the same.
co-ancestry. It's the
Yeah.
co-ancestry between breeders.
Exactly. Yeah.
It's the same idea between whatever you want to include in the error.
Exactly. Yes. And if you're explicitly modeling genotype by environment interaction, it's the same thing. So, you know, the mixed model equations, they're agnostic, if you would, to the type of effect. It's just, is it fixed or random? And if it's random, well, what's the covariance structure for those random effects?
Oh, thank you.
Okay, Yvonne, and then Fluture.
Hi, can you hear me?
Yes, I can.
Okay. I guess I was wondering with the A matrix, like when you do not include a marker matrix, what does that reduce to? Because I know that in some BLUP equations, people do not include the marker information and still obtain phenotypic BLUPs. So I'm wondering, how does that, what does that look like? Is it just nothing?
Okay, so yes, in this case, we used pedigree information. Okay, in this example. So if you recall the A made, I think the A matrix, if I recall, it was 2, 0, 1, 2, 2, 1. I think it was, I think this was our A matrix. And so this one includes, this is uses pedigree information. And the elements we said are equal to twice the coefficient of co-ancestry among the individuals. So each one is inbred, coefficient of co-ancestry of 1. That's why you have 2 in the diagonals. You can also, as we showed earlier, much earlier in the course, we can calculate Fij from marker information. So that means to say that if we were to calculate Fij based on marker information, it's possible that we might get something, and I'm just making this up, we could get something like 1.2. And then this could be, say, 0.8. And why 1.2 and 0.8? This means to say that each parent did not have an equal contribution to inbred 3. That based on marker data, we're saying, oh, parent 1 actually had a greater contribution to inbred 3 than did parent 2, and that's why it's not equal for these two. Now, if you ignore relatedness, or if there is no relatedness, then what you boil down to is you could just have a covariance matrix that is an identity matrix. And so you're ignoring relatedness here, and we see that because the off-diagonal elements are now all equal to 0. It turns out then that if all the data are balanced, the only fixed effect is equal to the mean, and everything is unrelated with each other, then the BLUP of an individual is simply equal to your heritability times the observation times the mean. And that's familiar, isn't it? I mean, it's very much related to, well, we did an exercise on this earlier, but it's related to, you see the shrinkage over here, shrinkage towards the mean. It's related to response to selection, because this would be your selection differential multiplied by heritability. Yeah. Okay, Fluture.
I think I have a more practical question.
Yes.
Because we always discuss when would I use blues as a basis for my further calculations, and when would I use BLUPs?
Mm-hmm.
We used to do a lot of GWAS, and then we discussed this several times, but are not very sure when we would use what. So
for GWAS, if you let's say you're doing GWAS but you have unbalanced data, I would use blues. I wouldn't do BLUP twice because, well, depends. So many of the GWAS approaches, they use a BLUP approach, sort of approach. And you don't, I think I wouldn't do BLUP once and do another BLUP on the BLUPs themselves. So I would do blues, best linear unbiased estimates, basically to get rid of the fixed effects. Okay, get rid of the fixed effects. After I've done that, then you could begin, then you'd apply BLUP. So you're basically, what you're doing is you're partitioning out. Instead of having one step, you're doing the fixed effects first, and then you're doing the random effects next, rather than doing BLUP and then BLUP again. Of course, there's really no reason, unless computationally or software-wise, to do it in two parts. I mean, you could do both simultaneously.
Okay, and then does that mean, because that's also, we were leaning more towards blues for the GWAS, but then does that also mean, so if you do genomic prediction, you would go for the BLUPs?
The genomic prediction would be BLUPs, but the observations that would go into your training data set would be blues.
Aha, okay.
Okay.
Yeah, yeah.
Does that make sense? Yeah, it's the same thing. Again, you have a training data set, can calculate the blues for those, and then you could do your training model, or calculate your, make your training model. Or you could do it all at once and have both the fixed and random effects for the markers.
Okay, but then when would you use would you then use the BLUPs only for when you really want to know the breeding value of the line?
Right.
Okay.
So I guess a quick answer, I wouldn't use blues as an intermediate step for further analysis.
The blues?
But I'm sorry, I wouldn't use BLUPs as an intermediate step for further analysis.
Okay.
Okay.
Yeah. Okay. Yeah. Thank you.
All right. There were two other hands that were raised. Go ahead. Nico had a first question. First of all, can you calculate the last part in the mixed model? Yeah, the fourth quadrant is, again, it's Z prime Z plus the inverse times VR VA. Basically, you just have to calculate this separately, which we did. And then you calculate this separately, well, which I showed. We didn't calculate it, I did it by computer. You multiply each element of this one by this ratio. And then you just add the two one-to-one correspondence addition of those two elements. Yeah.
Thank you.
Okay, who else had a hand raised? Francisco, is that a new hand, Francisco, or the previous hand?
Yes.
Okay, a new
it's
hand.
just related the question that.
And then Dietrich later on. Yeah, go ahead.
Normally in the paper you see two-step analysis. So the first step you have the blues, where you get rid of any variation that you see that rep, and then you get the BLUPs.
Yeah.
And then across locations, right?
Yeah.
And then you use that BLUP to get genomic selection on GWAS, right? That is something that you can do, or what?
You mean you use the BLUPs? You analyze the BLUPs as your training population?
No, you have 2 years for location, and then you get the blues for each environment, like particular environment, one environment, and then you get the BLUPs across all the environments. And then you use that value that you get rid of any variation that you have to get your estimation of GWAS or genomic selection. Is that correct? Or what could you recommend in order to this two-step process? Or my question is, how can you get rid of the rep variation, the reps, and then across the years, and then get the final value to use?
Yeah. So if you're doing a two-step, what I would recommend is step one. You obtain the blues. No, okay, sorry. what I would do is this. In step 1, I would do my mixed model equations to estimate B. Okay, is that clear? We estimate B. So in this case, we estimate the two B's. Step 2, I would correct my Y for my B, meaning that I would have my corrected Y is equal to my original Y minus X times B. Okay, I'm simply correct. I'm not doing blues. I'm not doing BLUPs here. I'm simply correcting each observation for its fixed effects. Okay, and then what I would do then in step 3 is I would use my corrected Y for whatever I want to do. I would use my corrected Y for my training population. I would use my corrected Y for GWAS. You'll notice that I'm not saying these are blues. So I'm not pretending that genotypes are fixed. And then later, oh, no, I just pretended they're fixed. Now they're random. No, I'm not doing that. Because I'm doing my full mixed model with fixed and random effects for genotypes, okay? Then I do my step 2. All I'm saying is, well, those fixed effects, they're just to correct for noise. And so therefore, I'm going to do a second step. I correct my Y for the noise that I have by Y equals Y, my corrected Y equals Y minus XB. And then it's this corrected Y that I would use in my GWAS or in my training population. So I'm not using my blues for GWAS or as in my training population. I'm simply using the corrected Ys. I hope I explained that well, but it's a key concept, I think.
Yeah, the B then will be reps, will be location, will be yield.
Yep. The B would be rep, location, years, transgenes, fertilizer, previous crop, whatever it is. Yeah.
Okay.
This would be XB. It should be XB prime here. Yeah.
Thank you.
You're welcome. Dietrich, you had a question.
It's not a question, a remark. Previously, somebody asked about how to perform these simple matrix calculations. I recently discovered it's easily possible in Excel as well. I put a web link in the chat. It was quite easy to do it in Excel for small data sets.
Well, thank you for that. I didn't know that. Well, I knew Excel you could do regression, for example. So you'd think that you should be able to do that. There's a way. So thank you for posting this, Dietrich. This is helpful. Just for the package, the package I use is a package called Scientific Word. Scientific Word. You can write documents, you could do math, and you could do matrix stuff in Scientific Word. This is not. I'm not saying you should buy it. I'm just saying that. Yeah. Okay, Fluturi had a new question.
Yeah, it then builds on what Francisco asked. So would you then suggest, do you think it's better than to use the blues for further calculations, or to do it the way you just described here with the 3 steps?
The purist in me prefers this one.
Okay.
The purist in me prefers this one because you are not making 2 opposite assumptions. You're not assuming that your lines are fixed, and then later on you might say, oh no, I'm going to obtain their blues, they're now random.
Yeah.
And so that's the main reason why I prefer this one here. Whether or not you get the same result or not, I actually don't know. I don't know if you do. If you get, you probably will get the same result. I would think you would get the same result in the end.
They should be at least very similar.
Yeah, at the very least. But again, it's more the purist in me, so to speak. Yes.
Okay.
Francisco.
Sorry, just I want to now.
Okay. Are there other questions from the group? If none, well, thank you so much for your engagement today. We'll continue this topic of discussion when we meet on Thursday. Have a good rest of the day, everybody.
Thank you.
Thank you.
Okay,
Bye-bye.
thank you.